Drew already took eco methods, so he's heard this before. So it'll be all review. Anyone here going to take eco methods?
I plan on graduating, so no.
Okay. That'll be fun. Okay, so what's bioassessment? This is when we use biological communities to evaluate the health of an aquatic ecosystem. You might say, why would you bother doing that? It seems like a lot of work. And it is. So the alternative to bioassessment would be to go out and take like a water sample and do a lot of the analysis that we did, like turbidity and nutrients, and can look for pollutants and things like that. And some limitations to that sort of analysis is that if you go out somewhere and just grab one water sample, that's kind of a snapshot sample, especially in streams that have flowing water, where you could, like, let's say you had someone dumping pollutants into a river at 3 in the morning, and then someone went and sampled at 8 in the morning, and you wouldn't get that toxin in your sample, but all the organisms have still been exposed. So just getting one sample one time is just kind of a snapshot. Doesn't really give you a broader picture. And if you sample organisms and look at the community, so if you think about what organisms are doing out in these ecosystems, they're just basically sampling the environment all the time. And so they're always being affected by the environmental conditions, and so they are able to kind of integrate all of that information. So bioassessment uses the organisms to tell us about the health or condition of the ecosystem, with the idea that they've been exposed to it their whole lives, and so the state that they're in tells us something about the state that the ecosystem is in. Make sense? Okay. All right, and just a fun term that's good to know, autoecology. So auto means self, right? So ecology, kind of ecology of the self, but it's really about species and what they require. And so when we understand the ecology of various species, we know like what conditions they tolerate, what conditions they prefer. So for example, rainbow trout like colder water temperatures. Down here it's naturally too warm, they don't survive well here. That would be an example of one little bit of their ecology. Even for species of algae, we know like what kind of pH they like, what kind of nutrient levels they like, what sort of temperature they like, all that stuff. So if we know that about a bunch of different organisms, and then we go sample and we get these organisms, then we can kind of like look at the organisms and then that'll tell us something about what the environment is like. And so some variety of things can be done in the bioassessment. There's a habitat assessment where there's one for streams that gets used. That's kind of there's 10 characteristics, and you rank them on a scale of, or maybe there's 20 characteristics. You rank them on a scale of 1 to 20, 1 being crappy, 20 being nice. And so you look at things in a stream like, are the banks eroding? Is there a lot of sediment in the stream? Is there vegetation on the sides? What's the land use around it? Are there root wads? Things like that. So that could be one question. And in bioassessments, we can look at organisms, and we need to understand the range of like the range of species that's there with their variable trophic positions and variable tolerances. So the organisms we were collecting and looking at understand that there's this range of where they would be in terms of trophic levels and what kind of environmental tolerances they have. And so macroinvertebrates are very commonly used in bioassessment. Fish also sometimes get used, and then algae also sometimes get used. I would say macroinvertebrates are the most popular group of organisms, and then fish, and then algae. And I think algae are just not used as much because there aren't as many experts on algae. Occasionally there's some vegetation bioassessments also, but those aren't used very often. All right. There's also some ecosystem measures that you could do. The productivity stuff we did off of the dock, you can do that sort of thing in stream as well, or you can in a lake, and so that could tell you something about what's going on in the ecosystem. Leaf breakdown, which is not something that we've done, but we talked about it a little bit. So you can basically get these mesh bags, and you get some dried up leaves and stuff them in there, and you know the weight of the leaves before you put them in the stream or the lake, and then you go put them in there, and you leave them for some amount of time, and then you collect them and get the leaf material out, dry it, weigh it again. You can see how much weight is lost, and that loss of weight is going to be from microbes feeding on it and inverts feeding on it. And then you can also collect inverts out of those mesh bags and see who's there feeding on them. So these take more time, but it's another thing that can be done, but it's biological. Okay, let's talk about fish and some pluses and minuses in using fish for bioassessment. So some advantages to using fish, they live a pretty long time, so they're pretty good indicators of sort of long-term effects, like from year to year or years to years. Fish might be a good option. Fish, compared to invertebrates and algae, they're relatively easy to identify as species. Anyone know that fish? I think we have it around
I
here.
should know, he used it recently.
He's like a
Is that a clown
Almost.
clown or rainbow.
It's a Halloween darter.
Yeah, that's right.
When I wrote this lecture for eco methods, like I was giving it close to Halloween, so I pulled up a Halloween darter. So when the male's in breeding, it has like black and orange on there.
We don't ever get to see them in color. It's always when they're dead and drained of all color.
Yeah. Gotta see them out in live. We know a lot about fish, the environmental requirements they need, their typical geographic distributions. We know a lot of that stuff for fish, because fish are, what we call the charismatic megafauna. So, everyone knows about fish. Oopsie. So there are some good things about using fish. There's also some disadvantages about using fish in bioassessment. So when you go sampling in the field to collect fish, it's a lot of work. There's a lot of equipment involved. So in a stream, you'll probably have the big backpack shocker, and you're going to be hauling all that stuff out to the stream and up and down the stream bank and walking around in the stream for a long time, pulling out fish, and Drew's like, yep, been there, done that. And so it takes a lot of work to sample a stream for fish. And also, most of the identification is done in the field, and so you have to have people on staff that can identify fish. So a lot of this bioassessment might be done by like state agencies or federal agencies, and so they've got to hire all these people to do this stuff. If you have questions about more short-term issues of water quality, like over a few months or even a few weeks, fish may not tell you something. So if you had if someone was suspicious that in the last month there had been like some kind of problem at the sewage treatment plant and they were letting out something harmful into the stream, the fish might not have, they might have been able to handle it for the past month or so, longer maybe not. So, but other organisms might respond more quickly.
Like algae?
Like algae or macroinvertebrates. See where we're going with that. And also, fish are not in all streams. There are some relatively small streams that don't have fish in them. In particular, the stream where I did my dissertation research, it was a perennial stream. It was always flowing, but it was so small, like this wide and this deep, and it just couldn't support fish. So fish would be useless if you were trying to do a bioassessment on that stream. And I don't know if Ruba talked about this when he talked about physical stuff with streams, but he talked about orders, right, first order, second order, third order streams. And most of our stream lengths are the first and second order streams. So actually most of our stream length in general is gonna be these smaller streams. So there's that. And then fish can run away when you're trying to sample them. So part of the fun with electroshocking is you've got someone shocking and you've got other people with nets trying to catch what gets stunned, and sometimes you don't get them.
We caught this one catfish that was too big for the net. It was a huge, and we got it with the electroshocker.
Mm-hmm.
It was huge.
I can see that. Those, yeah, catfish.
Garter are generally harder to catch because their scales are much thicker.
Okay, macroinvertebrates. Why would you want to use them in bioassessment? So they are quicker, easier, and cheaper to sample compared to fish. It takes much less time to sample invertebrates. You don't need as much equipment. It's easier just to carry what you need. It's not as much of an ordeal. Yes.
Would it be more like trapping than on-site collecting?
No, it would be, well, most of the time it would be on-site, and so there's a variety of methods. So there's the D-frame dip net, which the net that's about that long and shaped like a D, and you could drag that through the stream a certain number of times or in certain types of habitat. They have things like in the stream where I worked, they used a stovepipe core where it was this round metal tube with an open bottom, and you just jammed it into the bottom and you scooped up a certain depth and then took it all back to the lab and picked out all the bugs. So there's different ways depending on what kind of streams you have, what kind of habitat you have. But generally it's you grab it and you go. The traps that would be set, sometimes people do emergence traps, because a lot of these insects, as adults, they'll come out as flying terrestrial organisms. So you could have an emergence trap, and so you can catch them that way. And then another thing you might do is drift sampling, where you have a net that's about that big, water's flowing through it, and you just set it out overnight, and insects that might be stressed, like if there's some chemical in the water they don't like, they might let go of the bottom and then just drift away to try to go to a better place, or if there's predators around, they might let go and try to go to a better place. They usually do it at night because during the day it'd be easier to get caught by a predator. So, but that's not usually for bioassessment, but just general ecological info. Yeah, so in the field, quicker, easier, cheaper to sample compared to fish. And you generally get more species of inverts in any given sample than you would of fish. And more species equals more information. And macroinvertebrates are found in almost all habitats, even those small fishless streams. We have lots of macroinvertebrates. There's going to be something in that stream. And their life cycles generally run from about a year to maybe 2, and that seems to be a pretty good amount of time for integrating water quality. So fish that live a long time, they're better for longer periods. The past year or 2, if they're an older fish, they're kind of tough, been around for a while. The past couple of years may not affect them as much, but the inverts, they're going through their life cycles a lot faster, so we'll get more info. This little dude is a stonefly nymph, and we don't really have these in West Tennessee because it's too sedimenty here, but in Middle and East Tennessee we get these. Some are predators, some are detritivores, but Grubauer probably talked about stoneflies, Plecoptera. One of the sensitive groups of one of the sensitive orders of insects. Disadvantages for macroinvertebrates. So even though the sampling time is less out in the field, you've got to bring all those samples back to the lab and spend a bunch of time processing that and doing the identification. And so my postdoc that I had between graduating and coming here was at the EPA in Cincinnati, and that's a research group. Some parts of the EPA are more regulatory and some parts are more research. This was a research building, and they had a lab that did a lot of sampling in the summertime of macroinvertebrates, and then once the sampling season was over, all those people were in the lab picking bugs, which just means pulling the inverts out of the leaf litter or the gravel or whatever you sampled, and pulling all the bugs out, so the person identifying the bugs only had to look at the bugs. And so that's a lot of time to pick through all that stuff. Other disadvantages, there's some seasonal variability. So what you get in the spring may not be the same as what you get in the late summer. So that may kind of mess you up a little bit. Fish, you don't have that problem since they live so long. And let's see, macroinvertebrates don't necessarily respond to every pollutant. So, Grubauer's old advisor, Dr. Bruce Wallace, I believe he did a study in a stream where he dumped in a bunch of herbicide, see what would happen to the macroinvertebrates, and they didn't care at all. So the communities didn't change. So, or no, maybe he did the one where he dumped in insecticide, and then they all like drifted or died, and yeah, that killed them. But some study looked at herbicides, and they did not affect the inverts at all. So that's macroinvertebrates. And now algae. Why would you want to use algae? They're even easier to sample than inverts. You can just carry your toothbrush and your Whirl packs and your little field bag and just go in there and grab stuff and you're done even faster than the invert folks. Not always, but frequently there are more species of algae than there are of macroinvertebrates in a given aquatic system, and therefore more species, more info. And any place that's wet is going to have algae in it. As long as it's been wet for a few weeks, you'll get algae worth collecting. Another good thing about algae is since they're at the base of the food web, bottom of the food chain, they are that interface between the physical habitat and the biological. They're the ones directly responding to nutrient levels, for example. So they kind of interact more with the physical habitat a little bit more than the others do. Well, others still do, but algae more so. And algae have a pretty short life cycle, so they can respond pretty quickly to some change in environmental conditions. And in the summer, it might take about 3 weeks for ecological succession to take place and to reach a kind of that climax community. And you can do things like put out unglazed ceramic tiles and then come back in 3 weeks and see what algae have grown on them. There are these little rafts that hold microscope slides and they float. They're called diatometers. And you can put those out and come back in a few weeks and see what diatoms are growing on the slides. Anyway, and algae cannot run away. The macroinvertebrates, they're small, they can run away, but you can probably catch most of them. Algae, you're not going to be able to run away. So those are some pluses for algae. Disadvantages, there are none. Algae are perfect. I'm just kidding. Disadvantages, it also takes a lot of time to process samples and do the identification. So we have this thing called counting algae, and it's pretty common to get your sample, put it on the microscope, look in it, and you have to identify the first 500 cells that you see and put names on them. And so that can take hours.
What if you count the same?
So everything you count's going to be either pickled or preserved diatom slide. So you just go
I thought
don't backtrack.
it's you have used live.
Yeah, and they're like,
Yeah. So I'm what if you just keep counting the same one? It just keeps moving and you're
yeah, you
on
don't want to
side.
do that. Yeah, and diatom slides, they're permanent, so they're work pretty well. But yeah, it can take a fast sample would be probably 2 hours, average 4 or 5, but it can take a whole day to count a sample. So it takes a while. A lot of seasonal variability, so what you get in summer is going to be different from winter. And that's really all I could think of, yeah.
Well, I feel where fish you would have to have someone on the field that's knowledgeable.
Mhm.
I feel this you would have to have someone that's even more knowledgeable to properly identify the algae.
Yeah, you also need an expert and there aren't that many of them.
Yeah.
Yeah.
So I feel that would be a disadvantage.
Yeah, so most states that have some kind of bioassessment program have macroinvertebrate people and or fish people. There's only a handful that have algae, but Kentucky's one of them. Montana, I think Florida. There's probably more.
So would you be in high demand?
If they wanted to start a program, so that would be they may not want to spend money on it. fish versus inverts versus algae, like what should you pick? So if you have questions about geomorphology stuff, like habitat quality, just the like physical structure of the streams, you might want to go with fish. Inverts are pretty good for almost everything, so that's why so many people use them. And algae are good for nutrient issues, and for a while we've been talking about nutrient criteria in aquatic ecosystems at the governmental levels, and we know that we're adding a lot of nutrients, and so what amount of phosphorus and nitrogen is healthy, what's going to be too much, and so trying to sort out what's a good amount to have. And we're still working on that because it's not an easy question. Okay, so you've gone out and collected all these communities and you've identified species, and now you have this list of these species and how many of them you found. What do you do with all those data? Usually you calculate some kind of metric. And here's a big giant definition for metric: a calculated term, so there's a formula, or enumeration, just counting something, representing some aspect of biological assemblage, function, or some other measurable aspect. And this thing that you're measuring is a characteristic of the biota, and it changes in some predictable way with human influence. And I'll give you some examples on the next slide, but it'd be the kind of thing where you do a calculation, you get a number, and that number would tell you, oh, this is a really healthy stream, this is great. Or, oh my god, this stream is awful, it needs a lot of help. Or this stream changed from really good to really bad, something happened. So you get basically, it's like a GPA for your stream. And so examples of metrics, so for fish, even something as simple as species richness, you could use that as a metric. Darters are a type of fish that are generally a little bit more sensitive, so you could have darter species richness, proportion of individuals that are omnivores. So if you've got fewer specialists, more omnivores, that means your habitats may not be very diverse, and the only things that can live there are, they could eat anything. So the more omnivores you have, the more crappy your habitat is likely. And those are just some examples. Invertebrates, the percent EPT. Do you remember what EPT stands for? Rubal talk about that? These are 3 orders of insects, and it's Ephemeroptera, Plecoptera, and Trichoptera. So Ephemeroptera are mayflies. They're the ones that well, all of them have larvae in the water. Mayflies, when they emerge as adults, they live for a day or 2. They don't even have mouth parts. Their whole goal is to have sex, lay eggs, and die. And so they do that in about a day, and they tend to like pretty clean water, and there's often these mass emergences of mayflies where you get these huge swarms that come out in the summer over a period of a few days, and There's some places near the Great Lakes where they have to get the snow plows out to like plow them off the streets, like they can be pretty big.
It's also really bad around Duck River. We had to spray around the dam, and the person that'd be outside spraying would just be covered head to toe in mayflies.
Yeah, and but that's a good sign. That means the water is fairly clean. So, it's kind of like, bugs, but it's a good thing. Plecoptera stoneflies, I showed you a picture of one of those, that's that one. And then Trichoptera, the caddisflies, those are the ones that build the little cases that they wear, or they might they might spin nets to filter feed. And so all three of those, they all have like a few species that are really tolerant, but if you have a lot of different species, that's a good sign. And so EPT, percent EPT, so what percent of your community are those more sensitive taxa? The higher it is, the better. Do EPT to chironomid ratio. Chironomids are more tolerant to junk, and so, but still the higher that number, the better they are. Percent contribution of the dominant family, so that gives you a sense of evenness in the community. Those could be examples. Algae, you might do percent cyanobacteria, which they like high nutrients. Percent modal diatoms, so these are diatoms that can crawl around in sediment, and that's often an indicator of how much sediment is out there. And then the percent sensitive diatoms, so how many sensitive species do you have there? So that's some math that you can figure out. And one of the things that I did at my postdoc at the EPA was try to develop metrics to use for diatoms from streams on the Atlantic coast. And that was very pleasant, because it's just like grinding numbers, and I was like, oh my gosh, I can't take it. So those are all individual metrics. We frequently take a bunch of metrics and stick them together. And so we have the multi-metric approach, where you have a bunch of metrics and you lump them together, and you end up taking all those numbers and getting one number. And the ones that we typically hear about are called IBIs, or an index of biotic integrity. And you might see like FIBI or MIBI. We often say periphyton IBI for algae instead of just diatom, because you might look at more than diatoms. Phoebe, maybe, Phoebe. And you might even like use all of these organisms and have another IBI that like puts it all together. So this one is something from North Carolina that was an IBI score, like for the stream. I think it used inverts and fish. So you can do all kinds of math, but those are pretty common to see in the bioassessment literature. Okay, so that's bioassessment. I want to mention a little bit about something called paleolimnology, which can be used in bioassessment, but from communities in the past. And this is looking at historical environmental conditions, mostly in lakes. And here's what goes on. Here's a lake. That's the lake bowl. Here's your water level. And so in here in the water, you've got organisms that are living their little lives. You've got stuff blowing in from around the lake, landing in the water, and then stuff lives and dies. It might get decomposed, or it might just fall to the bottom, and you get a little tiny layer of sediment that year. next year it happens again, you get another little layer of sediment, just dead things accumulating. And so every year you've got another layer, another layer, another layer. So lakes are basically all like filling in slowly. And what's in all these layers are things like chironomid heads. So these little, that's a little chironomid. They have these little hard head capsules, and the body's all squishy, so that usually breaks down pretty quickly. But those little head capsules remain, and people that know about these things can identify those as species. So you can nerd out on chironomid heads if you want to do that. Pollen grains fossilize really well, so they would be basically terrestrial plants that blow in from outside, land in the water, and then fall to the bottom. And diatoms and chrysophytes that have the glass cell walls, they preserve pretty well. And sediment too, that silica, that glassy cell wall doesn't really break down very fast. So you can so what they do is, often they go out in the wintertime when the lake is frozen and you can walk on it or drive on it, and they go to the deepest part of the lake and they put a hole in the ice and they drop a coring tube down there, and then they pull it up and they've got this core of sediment that's all intact. And then they can, like that, and then they can put ages on the layers so they know that these are the youngest layers of sediment, these are the oldest. They can do some radiometric dating and get actual dates on these things. And so you can know when, like you can identify some organisms that were there, and you can know when they were there. And you can go back as old as the lake is, or as deep as you can core to get these get this information. And they used diatoms in a paleolimnology kind of way, looking at lakes in upstate New York back in the late 70s when acid precipitation was a big issue, because there were some lakes that were naturally kind of acidic, and then other lakes that were not naturally acidic but became acidified from acid precipitation. And they wanted to know which lakes were not supposed to be acidic so they could do some kind of mitigation and like throw a giant tomes in the lake and make it less acidic. And so the diatom folks, they figured out a formula basically based on diatom species and their abundances, and they could figure out the pH of that water based on the diatoms that they found out of the core. So that's kind of cool. And this lake, so back before 1960, the pH of the lake was around 6 point something, and that pH dropped to 4.5 by 1979. So that was quite impacted by acid precipitation. All right, good stuff. Groovy, love it. Okay, I think that's all. Yeah, this is what I did at the EPA. If you are interested at all in federal work, they use a lot of acronyms. And so the research division, it's not a division, the research lab, NERL, National Exposure Research Lab, they have a bunch of divisions. I was in the Ecological Exposure Research Division, EERD. We still have a little fleece that says EERD on it. We were in a branch, so this is kind of like department and college and university almost, except there's no students. And so this ecological research branch, we were, and I'm not going to touch you on this just for your curiosity. So what I was working on was everybody worked on standardizing methods for sampling, for bioassessment and biomonitoring, so that we could give out guidelines, and so everyone would sample inverts the same way or fish the same way, because then it would be easier to compare one group to another. I worked on developing paraphyte and IVIs, and then when I left 20 years ago, there were some new ideas about endocrine disrupting compounds or using ecosystem function measures. And there's the really ugly building where I worked. It was really ugly. It was weird, you had to like wear your little lanyard with your ID on it, and that's how you could get into the parking lot. It was strange. Okay, so that is everything I had to say about bioassessment. And everything I have left to say to you guys. Until